So yes, I'm going to be talking about nutritional influences on arsenic metabolism, predominantly in Bangladesh, but I will try to touch on the work of others that's been done in different areas. And this has come up a few times already in previous talks. And so I hope I can fill in some of the gaps and answer some of the questions that were raised. Okay, just I have no conflicts to report, and much of this work is funded through NIH grants. Okay, so I work predominantly in Bangladesh, which has probably one of the worst arsenic problems in the world. The water that comes out, so people in Bangladesh get their water from tube wells. And actually, prior to 1960, people in Bangladesh mainly drank surface water, which was a big problem because there was lots of bacterial contamination. There was very high infant and child mortality. UNICEF and other NGOs went in the 1960s and 70s and installed thousands of these tube wells throughout the country. This was great in that it provided drinking water that was free from bacterial contamination. It was not so good in that, about 15 to 20 years later, it was discovered that much of the water that is accessed by these tube wells is contaminated with naturally occurring arsenic. And so they have a very serious issue on their hands. So I just wanted to give you a little bit of background about Bangladesh. So this is a map showing the distribution of arsenic concentrations in the wells throughout the country. And you can see that the problem is most severe in the south of the country. It's less of a problem in the northwest. And we work here, and when I say we, so Colombia has a large ongoing project in Bangladesh centered around arsenic exposure through drinking water. There are many different facets of that project. There's a very large prospective cohort study that's been going for almost 15 years now, and it's up to about 40,000 participants who are followed up every 2 years. Their arsenic exposure is reassessed, and they are followed for numerous health outcomes, cancer outcomes, cardiovascular outcomes, diabetes, et cetera. And then there are several projects, including mine, that are nested within that larger cohort study and draw on participants from that cohort study. So we work here, which is about 30 kilometers east of the capital of Dhaka. And this area was chosen because, as you can see, there's a wide range of exposures in that area, allowing investigators in our group to look at dose-response relationships and to try to get at that important, less than 100 ppb exposure. In Bangladesh, so in the US, as has been pointed out, we have the 10 microgram per liter guideline. In Bangladesh, they are still using the old guideline, 50 micrograms per liter. Okay, so we know that arsenic is associated with numerous adverse health effects, several types of cancers, vascular disease, of course, pulmonary disease, and cognitive deficits. In the area in which we work, there are several efforts to reduce arsenic exposure among the people who participate in our study and among other people who live in that region who choose not to participate. One of those is through installation of wells that access the deeper aquifer that has lower arsenic concentrations. And our group, our group has installed several safe wells for participants to drink from. There are also filters in use. There are several different filter varieties available in Bangladesh, but I'll tell you that these filters do not work for very long, contrary to what the companies making them would have you believe. We used these in a study, and they lasted about 2 to 3 months, and then they were pretty ineffective. So that's an issue that we're exploring further. So one of the other approaches, and education, of course. Our group does a lot of education, teaching people about arsenic, why it's bad for you, why you shouldn't drink water that has high arsenic in it. And so I'm going to focus mainly on what we've done. In Bangladesh, the predominant exposure is, of course, through drinking water. We have focused less on arsenic from food, although I did want to point out that we recently have a publication showing that intake of rice, in particular steamed rice, is associated with urinary arsenic concentrations. We've also shown that steamed rice intake is associated with risk for skin lesions, particularly among people with water arsenic exposures less than 100 micrograms per liter. So in Bangladesh, the food exposure sort of becomes more important among people with lower water arsenic exposures. And of course, rice is absolutely the staple food in Bangladesh. It's eaten every day at every meal. So the water that comes out of the wells in Bangladesh contains predominantly arsenite, so the trivalent inorganic form, very toxic. And I just wanted to review a little bit about arsenic methylation, and then I'm going to tell you how nutrition comes into play in influencing arsenic methylation. So this arsenite can undergo a methylation step catalyzed, of course, by arsenic methyltransferase with S-adenosylmethionine, which I'll just call SAM for short, as the methyl donor here. So arsenite is methylated to the monomethyl pentavalent form, which can be reduced to the trivalent form. And you can see the methyl group that was added, which came from SAM. There can be another reduction and a second methylation step. So the monomethyl form is methylated to the dimethyl pentavalent form. And you can see there are the two methyl groups there. So there's a lot of interest in arsenic methylation, and why do we even care about arsenic methylation? Well, we know first that people differ substantially in their ability to methylate arsenic. And this is probably due to a lot of different factors, polymorphisms in arsenic methyltransferase that influence enzyme activity, nutritional factors, probably many other factors. So this is just a frequency distribution from some of our data in Bangladesh. And you can see that the green is the dimethyl arsenic, red is the monomethyl, and the purple is the inorganic arsenic. So some people, and these are urinary measures, so some of our study participants have, for example, between 80 and 85% of the arsenic that is in their urine is present as the dimethyl form, whereas other participants have between 50 and 55% as a dimethyl form. So that's a substantial difference. We know that the monomethyl form, in particular the trivalent monomethyl form, as has been noted by the other speakers, is the most toxic. And that's known based on experimental data, but also the epidemiologic data are consistent with that. So this is a summary of a number of studies that have looked at arsenic exposure in relation to a number of different health outcomes, several cancers, ischemic heart disease, skin lesions. And what these studies have done is to measure arsenic in urine, to measure the different metabolites in urine. And you can see that these are comparisons of high versus low urinary monomethyl arsenic and the relative risks of the various arsenic-related diseases. And that across the board, and of course, it's rare to see something this consistent, but when you compare people with the high MMA in urine versus low, they have increased risks of a number of diseases. Now, if you make a plot like this, I didn't show it here, but if you make a plot like this using the percent dimethyl arsenic in urine, high versus low, you see the opposite. What you see is that the point estimates tend to fall less than 1, so decreased risk of these various health outcomes. So we care about methylation and what influences it because we know that people differ substantially in their ability to methylate arsenic, and we also know that the arsenic metabolites differ in their toxicity and in their relationships to disease outcomes. Okay, so I mentioned earlier when I was reviewing sort of the arsenic methylation pathway that the methyl group for the two methylation steps comes from S-adenosylmethionine, or SAM. Well, so you can see SAM here. And where does SAM come from? Well, SAM is generated via one-carbon metabolism. And this is, I'll just say, a very simplified version of this pathway. It's actually much, much more complicated than this. There are many more arrows that come into play, and there are lots of long-range inhibitory or stimulatory interactions that take place. But just to give you sort of an overview or review, if you're already familiar with this pathway, essentially what happens is that dietary folate, so folate is a B vitamin, it donates a methyl group for the remethylation of homocysteine to methionine. This is a critical remethylation reaction. Vitamin B12 acts as a cofactor for this reaction, a cofactor for methionine synthase, which catalyzes this reaction. This methyl group gets passed along the pathway to methionine, which gets activated to SAM. And I should say that SAM acts as a methyl donor. It's sort of a universal methyl donor. So there are over, I think it's now, there are over 100 different methyltransferase enzymes that use SAM as a methyl donor. So arsenic methyltransferase is one of these enzymes that uses SAM. Also, DNA methyltransferases use SAM. So in this methyl group gets passed from SAM to a number of substrates, one of which is arsenic, and you can see the dimethyl form here. When folate is low, so when there's folate deficiency or vitamin B12 deficiency, homocysteine goes up because it is not, the cells are not able to efficiently remethylate homocysteine to methionine to generate SAM. We know in Bangladesh that there is a lot of hyperhomocysteinemia, so a lot of very high homocysteine concentrations. I'm not going to show the data here, but just to tell you that is absolutely true. We've looked at it. There's also a lot of folate deficiency in Bangladesh. Approximately 2/3 of males are folate deficient. 2/3 of males also have high homocysteine. And a substantial number, about 35% of females are folate deficient, much less than males. Part of the difference is probably related to the fact that men in Bangladesh smoke cigarettes a lot. There's a lot of smoking in Bangladesh, which can also interfere with folate and destroy folate. The women traditionally don't smoke, although they do chew betel nut, which can also influence folate concentrations. Okay, so that's just sort of setting us up for understanding how nutritional factors might come into play. I'm going to focus mainly on the factors that we know, the nutritional factors that we know are involved in one-carbon metabolism and in that generation of SAM. Other groups have looked at other nutrients that are not involved in that pathway. I'm not going to talk about those today, just for the sake of time and sort of unifying the presentation. So our group, and in particular Dr. Mary Gamble, started this research on folate and how it might relate to arsenic methylation. And she knew from a number of cross-sectional studies she had done that plasma folate levels were related to the percent of each of the urinary arsenic metabolites. So measure folate and urinary arsenic metabolites at the same point in time, look statistically at associations between them. Folate was positively related to the dimethyl form and inversely related to the inorganic arsenic and the monomethyl form, suggesting that folate was favorably associated with arsenic metabolism. She followed that up with a randomized controlled trial of 200 folate-deficient Bangladeshi adults who were randomized to receive either folic acid or placebo for 12 weeks. And blood and urine samples were collected at baseline and at different points throughout the study and then at the end of the study period. And what you can see is that individuals who received folic acid, so the solid line, had an increase in the, a decrease in the percent inorganic arsenic in urine over the course of the study, a decrease in the percent MMA, and an increase in the percent DMA, suggesting that in these folate-deficient individuals, folic acid supplementation for 12 weeks helped to improve arsenic methylation capacity. We had some blood measures. You can see that blood arsenic went down about 14% in individuals who received folic acid, and just a very small, not statistically significant change in the placebo group. I can tell you that this decrease was predominantly a decrease in the percent of the monomethyl form, the trivalent plus pentavalent. We can't, I should say, when we measure the arsenic metabolites in urine and blood, we cannot distinguish between the trivalent and pentavalent because after we collect the samples, they're easily interconverted, so we can't make any firm statements about that. So we express it as percent inorganic, meaning trivalent plus pentavalent, and percent monomethyl, trivalent plus pentavalent. And here again, we have a frequency distribution. At the baseline of this study that I just presented, you can see this is the percent dimethyl arsenic in urine at baseline. And what happens is that after 1 week of folic acid supplementation, a distribution is already shifted to the right, and after 12 weeks, it's again shifted towards the right. So folic acid supplementation shifts the entire distribution of dimethyl arsenic in urine. Now I wanted to come back to touch on vitamin B12. There's been less interest in vitamin B12 than there has been in folate. I'm not really sure why that is. B12 is absolutely an essential cofactor in this pathway, and you cannot, the methyl group that comes from folate gets stuck in this cyclical pathway here unless you have B12 to pull it forward this way. So we took a look at vitamin B12 and its relationship with arsenic methylation. We looked at the literature first to see what had been done. There's not too much out there on vitamin B12 despite its importance in the one-carbon metabolic pathway. There's some dietary data showing that dietary B12 intake inversely associated with percent inorganic arsenic in urine and positively associated with the percent monomethyl form. No association with arsenic-induced skin lesions in a study by Chang et al. In 2006. Another study showing no association with arsenic-induced skin lesions. And then some non-significant associations inverse with the percent inorganic arsenic in urine and non-significant positive with the percent of the dimethyl. So we took a first look at this using a cross-sectional study. We measured total vitamin B12 in plasma, and we measured arsenic metabolites in urine. And these are parameter estimates, so beta estimates from regression models in our total sample. So you can see that vitamin B12 was inversely associated with the percent inorganic, positively associated with the percent monomethyl, and absolutely no association with the percent dimethyl, which is a bit concerning. We stratified to look to see if there was a modification of this effect by folate status. And really, all of what's going on here is in the folate sufficient. So what this is saying is that if you don't have adequate folate, then there's really no influence of vitamin B12, right? Because you need initially that methyl group from folate, and then vitamin B12 can come into play. So our data are consistent with what we know about the metabolic pathway. Our group has also done a nested case control study of arsenic-induced skin lesions. So this is nested within that larger cohort that I was telling you about. So we took all of the skin lesion cases that developed within the first 5 years of the prospective cohort study. We matched them to controls based on age, length of follow-up, and water arsenic exposure, water arsenic dose. And we asked then, given a particular, water arsenic dose, what's the relationship of these nutrients, so B12 and folate, and what's the relationship of having high homocysteine with risk for skin lesions? So the finding for vitamin B12, so this is low vitamin B12 versus normal, and there's some increased risk, although it does not reach statistical significance. There's a significant positive association for having low folate and again, a significant positive association for having high homocysteine, suggesting that these nutrients are important for arsenic methylation. In particular, folate, vitamin B12, I'd say we need much more work in this area, and that there are some potential adverse health effects of nutrient deficiencies in this arsenic exposed population. This is data from Taiwan. I'm sorry for the busy slide, but this is a case control study of urothelial carcinoma, so bladder cancer. And they looked at total arsenic exposure in relation to risk for this outcome, as well as the percent of the arsenic metabolites in urine, so percent inorganic, percent MMA, percent DMA. They found strong positive association between total arsenic and risk for bladder cancer, and positive associations with the percent inorganic and the percent MMA, and an inverse association with the percent DMA. They also measured folate, plasma folate levels, and they found this very strong inverse relationship, dose-response relationship, between folate concentrations in plasma and risk for this bladder cancer outcome. They went on, I'm not going to show you the data, but they went on to look at, statistically, to look at interactions between the arsenic metabolites and folate. And they did find interactions suggesting that folate became even more important when there was higher arsenic exposure. So much of our work has focused on folate for perhaps obvious reasons. It plays such an important role as a methyl donor in this pathway. But we've become interested, in particular, I've become interested in what is the role of other nutrients in this pathway aside from vitamin B12, and can we get a better understanding of how these nutrients might interact with each other to influence arsenic methylation. And so we're starting to explore this area. So I told you that homocysteine can be remethylated to methionine with a folate group, a methyl group from folate and vitamin B12 acting as a cofactor. But there's another way in which this remethylation reaction can happen, and that is using a methyl group from the nutrient betaine, which is catalyzed by this enzyme here, BHMT. You can get betaine in the diet, or you can get betaine from the oxidation of choline to betaine. Choline can also be obtained in the diet, and it actually can be synthesized here using SAM as a methyl donor. So it's an interrelated, it's a complicated pathway, but both of these can come from diet or they can come from endogenous synthesis. So essentially what happens is when you have a lot of folate deficiency, betaine becomes more important as a methyl donor for that remethylation reaction. So again, we took a first look at this in some study participants in Bangladesh. We measured, in collaboration with Marie Caudil at Cornell University, who is sort of an expert on choline and betaine metabolism and has the technology to measure those nutrients in her laboratory. We measured plasma concentrations of choline and betaine. We looked at their relationships with the arsenic metabolites in urine. And we looked stratified by folate status. We want to see, is what we hypothesize consistent, are the results consistent with what we know about the metabolic, one-carbon metabolic pathway. So if you look in the total sample, you see that betaine is inversely associated with inorganic arsenic in urine and percent monomethyl in urine, positively associated with percent DMA. The relationships are less strong for choline. When you stratify by folate, there's some suggestion of the same relationships for betaine, but they don't reach statistical significance. When you look in the folate deficient, there you really see strong effects of choline and betaine, so suggesting that they are favorably associated with arsenic methylation. And this is an area that we are taking further, and we're doing some, I'll tell you a little bit later, but we're doing some pilot studies looking at choline and betaine supplementation in Bangladesh. And this is just to show you what the plots look like. So these are for the folate-sufficient and the folate-deficient, plasma betaine versus urinary percent dimethyl arsenic. And they're modest relationships, but they're definitely there, and again, stronger in the folate-deficient. Okay, now other groups, I have to say there's not, the literature on nutritional influences on arsenic methylation is not, it's developing. It's still somewhat limited. Other groups have taken a look at this. Alan Smith's group from Berkeley conducted a cross-sectional study of about 400 people in West Bengal, India. They looked at a number of dietary factors. They had a dietary questionnaire to assess intake on, they probably looked at 30 different nutrients. They had plasma measures on some of them, urinary measures on some of them. Their main finding, I've just summarized their results here, their main finding, they found that folate was associated with increased percent urinary monomethyl arsenic, as we have and as others have. And they also reported that urinary creatinine is positively associated with the percent dimethyl arsenic. Now, for those of you who study arsenic, this is complicated. So what is urinary creatinine? It's a breakdown product of creatine. Creatine is used to regenerate ATP in muscle. Creatinine is excreted at a fairly constant rate in urine. And for that reason, it is used as a urine dilution adjustment factor in studies of arsenic. So when we measure urinary arsenic, we express it as per gram creatinine. So the fact that urinary creatinine is related to arsenic methylation, and I can tell you that we've seen this in a number of independent studies, and other groups have also reported this, suggests that adjusting urinary arsenic for urinary creatinine is probably much more complicated than it seems. And it gets even more complicated because creatine, the precursor of creatinine, is actually synthesized via one-carbon metabolism with SAM as the methyl donor. That consumes about half of all available SAM. The synthesis of phosphatidylcholine, which can be converted to straight choline, consumes about 40% of all SAM. So these relationships are really complicated, and trying to tease them apart has been somewhat challenging. So I know I'm short on time, so I'm going to try to go on. Just to let you know what we're working on, we have a new study that is underway looking at folic acid supplementation in individuals who are not necessarily deficient for folate. So we've taken a general sample of our population in Bangladesh. We're also doing some work with creatine supplementation. We have some thoughts about how creatine might be related to arsenic metabolites. Basically, the idea is that supplementing with dietary creatine will free up methyl groups for the methylation of arsenic. We don't have results from this study yet. Samples are being analyzed, but we're excited to see the results. And here's just the pilot study of supplementation with phosphatidylcholine and betaine that I mentioned. Again, with the outcome of arsenic metabolites in urine and also in blood. Additionally, to try to tease apart how all of these nutrients interact with each other, we are collaborating with 2 investigators at Duke University to do some mathematical modeling of arsenic methylation, which the mathematical models are essentially based on enzyme kinetics and work through a series of differential equations. And they're allowing us to ask questions in silico about how nutrients might be related to arsenic methylation that we can then test in our population in Bangladesh. And we're also very interested in the role of glutathione in arsenic methylation, and this comes back to the one-carbon metabolism pathway because the synthesis of glutathione is linked with one-carbon metabolism through the transsulfuration pathway. And there are a number of dietary factors, cysteine, glutamate, which go into the synthesis of glutathione, and so we're looking for possible avenues of intervention there. I think am I out of time? Yeah, so I'm not going to talk about children, but thank you. Happy to take questions.